Okay, so lab this week goes over what's called tonicity, which is kind of the movement of water as it's related to the solutes that are surrounding it, or within it, I should say. So in this thought experiment, we have an equal amount of water in two sections that can pass either way through this membrane right here. Now if we add a solute, like let's say salt, and we have it in pretty equal proportions on each side, the water's not going to change. The thing is, if we add too much salt to one side, everything's changed because the salt can't cross this membrane right here. Only the water can. So naturally, if this is, section 1 and section 2, if there are more solute molecules over here, water behaves to try and make the concentration of the solutes equal. So in this case, these are not highly concentrated, these are highly concentrated. This solution is hypertonic. It has too many solutes, thus hyper. So in this case, the water will exchange because the salt can't. And essentially, you lose a lot of your water here, goes down, and a lot of it goes up here. So now we have all this water in here that dilutes these to this same level of dilution. So essentially, water will move towards solute in order to make the entire thing the same concentration. The reason it does this is because this membrane right here, water isn't affected by that. It can pass through either way. Yeah, don't mind the physics of the membrane like falling on itself. You'll see where we're going with this. So in a human cell, you can say a blood cell, there's water inside of it, and for the sake of this experiment, infinite amounts of water outside of it. Because if you think about the body, the cell is so small compared to the entire thing, there's all this water out here, but it's at the same concentration, it's fine. And there's a certain amount of solute, and let's say salt again. So right here, I'm going to show what isotonic is. Isotonic means that this surrounding extracellular area has the same amount of little solute molecules, and thus the same concentration of solute as the cell does. So this is very important that your body keeps these ions, such as salt, in very good proportions. Because if you don't, so right now we're isotonic, which means solutes are even, and the water isn't moving anywhere. It can move in and or out of the cell, doesn't matter. Let's say you're in the middle of the ocean and you take a huge drink of salt water, all of a sudden, you have a hypertonic solution. The reason this is a bad idea, among other things, just being in the ocean in general is terrifying. The other problem is that this is hypertonic now, out here. Because there's too much solute. Now, solute, it can't cross the membrane to even out the concentration. It gets rejected. However, the water is going to try and make the concentration, it's going to try and make the concentration in here, which is a lower concentration. It's going to try and match this. The problem is that the hypertonic solution is going to cause the water inside the cell to move out, because the water is trying to dilute this hypertonic solution outside the cell by pouring its water from the cell into the solution. The problem is that this causes the cell to essentially shrink, because now all of its H2O has been donated to the hypertonic area out here because it was trying to dilute that salt down to the normal levels, but it couldn't. It's not happy and it's probably dead. Even worse is something called a hypotonic solution. So in the case when your cells, and say this is just normal right here, the amount of solute here is very high compared to what's outside, that's not good either. Because remember that there are infinite amounts of water outside the cell that are surrounding it. And if it's not isotonic, so see in this case there's very little solute here, water will try and move into the cell to correct this concentration. This concentration's too high. See how much red solute's in there? It's gonna pour into the cell. And animal cells, they're just gonna burst. And they burst open because the water pops them open. Plant cells are much more rigid. They have this nice like cell wall, and they just kind of like expand and stretch. That's why plants can stand on themselves, kind of. So in that case, the cell was in a hypotonic solution. Hypo, because there was very little of the solute. So the water from here tried to mimic the concentration, and the H2O out here burst the cell. Boom. Again, the cell is now dead. The whole idea behind this concentration and these experiments, it's not the easiest thing to grasp why chemicals move this way, but it's kind of just a basic inherent rule that concentrations and water as a solution tries to make all the concentrations always fit and always be the same. So there's always a danger in messing with solutes. That's why, you're not supposed to eat tons of salt. You're not supposed to drink salt water, things like that, because that messes with everything.